Hi class, this is Sir Alex Basco and welcome to our class in Biology 1. Today we'll be discussing our lesson 5.1. Today we'll be talking about cell cycle and mitosis. So what are our goals for today's lesson? Today, at the end of the lesson, I want everyone to be able to characterize the phases of the cell cycle and their control points, as well as be able to describe the stages of mitosis. So there are two things that we will be discussing specifically today. One is cell cycle, and more specifically, which is part of cell cycle, is mitosis. So cell cycle and mitosis. You can think of cell cycles as the life cycles of the cell, and it usually starts the moment that the cell was given parang nabuo siya via mitosis, of course. Cells can only be reproduced via mitosis. So after the parent cell divides into two daughter cells, the daughter cell then starts to working for it to divide again, and that's how the cell cycle starts. It starts its growth, and then until from the moment of its birth until it reproduces. So the cell cycle is between its conception until it conceives or it creates another cell. For a cell to divide, there are two important things for it to do. One is for it to physically grow. It is very important kasi kung maliit masyado yung cell, it is difficult for it to parang basically separate itself into two complete cells. And then another thing is it needs to replicate its DNA. So these two things are very important for the cell to achieve first before dividing itself. Cell cycle and mitosis is very organized and predictable. This is very important, especially for the replication of DNA. Kasi if this division of cell is not organized in a way to ensure that the cell that it creates, its two daughter cells, will have equal DNA and all have the parts it needs, mamamatay lang yung cell. Or if it has too much DNA and the other one have too less DNA, pareho pa rin most likely mamamatay yung cell. That's why cell cycle and mitosis is very organized and predictable. It needs to be that way for the cell to survive. And there are two major phases of cell cycle. One is inner phase, and the second is the mitotic phase, which we'll be calling M phase from now on para mas madaling yung discussion natin. So inner phase and M phase. So here's a summary of the cell cycle. So this whole part right here, this purple, green, and pink, are all part of inner phase, which means for most of the life of the cell cycle, or for most of the time of cell cycle, the cell is spending most of its time growing, duplicating DNA, and preparing. Kasi ang cell division nangyayari lang siya sa mitotic phase or sa M phase. So we call this mitosis or cell division. On the inner phase, cell growth, DNA duplication or replication, and cell preparation for mitosis are happening, and we call those gap 1 or G1 S phase and gap 2 or G2 phase. Okay, so I hope that's clear. Now let's talk about inner phase, and like I mentioned, there are three phases in inner phase: G1, S phase, and G2. So first, let's talk about G1 or G phase, or gap 1 phase, or also it's called gap 1 phase. So during this phase or G1 phase, the cell physically grows. Moreover, it copies its organelles. That's why it grows. Kasi, if you guys remember what organelles are, these are membrane-bound parts of the cells inside it or in the cytosol. So like the mitochondria, like the lysosomes, the Golgi complex or Golgi bodies, the vesicles, Lahat ng yon, dinuduplicate, ini-start ng i-duplicate ng cell. And for it to house the double amount of organelles inside it, it has to physically grow. Next part is S phase, or what we call the synthesis phase. So synthesis means to create. So what we're doing here is we're creating DNA copy. So at S phase, the cell is spending time, is spending energy to create a copy of its DNA. Moreover, during this phase, centrosomes are also duplicated. Later, we'll discuss more ano bang ginagawa ng centrosomes. But these are very important structure for organizing the microtubules that the cell will use to divide. So it has a very important centrosomes, or has a very important tasks in cell division. In G2 phase or gap2 phase, what happens is the cell continually grow. Moreover, it creates proteins and organelles. Like I said, it grows because it starts to create more and more copies of its organelle and more proteins for the cells, for the two new cells to use. Also, it organizes, at this phase, it organizes the contents na. So, meaning, dahil doble na yung amount ng organelles, you have twice the amount of mitochondria, twice the amount of DNA, twice the amount of uh, endoplasmic reticulum, twice the amount of lysosomes. So, ang ginagawa na ng cell, it starts to organize it in a way na kapag nag-divide siya, both of the cells na magiging daughter cells, they would have the equal amounts of the organelles and the equal amounts of DNA, na and yung laman nila is the same as yung laman ng parent cell. Okay, so it's basically duplicating itself, and all its contents is still similar. Okay, I hope that's clear about G2 phase. And G2 phase ends when mitosis begins. One important checkpoint, so ito yung kanina part ng goal, the checkpoints is the G2M checkpoint. If the cell, before the cell continues to mitosis, which is basically the cell division, it has to undergo a checkpoint. This checkpoint checks whether there are damages in the DNA or whether damages in the organelles. And what the cell do is it has to repair all those damages. Otherwise, it will not continue in mitosis. Okay, this is very important kasi nga, if, especially on the DNA, if there's too much DNA, or if the duplicated DNA is damaged, it has to be repaired first. Otherwise, yung magiging duplicate niya, yung magiging daughter cell niya, it will not survive. Okay, so if there are damages, the division will stop, and the cell will spend time repairing those damages. Next, mitotic phase or M phase. So M phase is the start of cell division. M phase involves two distinct division related processes. So there are two distinct division processes in M phase. One is mitosis and the other is cytokinesis. Okay? Although when we talk about mitotic phase, usually we interchangeably use it with mitosis. But mitosis is a very specific process inside M phase. And another part of the process of mitotic phase or M phase is cytokinesis. So we'll talk more about mitosis and cytokinesis later. But for now, let's talk more about M phase. Ano ba nangyayari sa M phase? Oh, here. So mitosis is the majority of the cell in the human body undergoes mitosis. So mitosis, this is basically the like I mentioned earlier, it is used interchangeably with my mitotic phase. And most of the cells in the human body undergo mitosis except for two important cells, which are the gametes or the sex cells. And mitosis has the following functions. So why do we have to undergo mitosis? One is for growth and development. So the only way for a person to grow from baby to an adult is for its cells to continually divide. If the cell is not dividing, you will not grow. But do not think of in a way then. So like in a way then, kung mas short yung isang tao in terms of height, they basically have less cell division. And that's, you can blame that on your DNA or your genes kung bakit mas shorter yung tao. But growth and development, yeah, is part of mitosis. Next is replaces worn out cells. So as the cells continually to function and grow, of course, there will come a time na mamamatay sila, called the apoptosis. And when the cell dies, it has to be replaced. Okay, it has to be replaced by the other living cells. So meaning they have to continually divide to cover out that worn out cells. And they do that via mitosis. Another is increase the population for single eukaryotic cells. Example, yeast. Yeast a single cell eukaryote. It means its whole body is just one single cell. And in order for it to survive, it has to duplicate itself to create, let's say, a family, kumbaga. Tayo, yung cells natin nagdi-divide for us na singularity, na parang singular person na tayo. Pero kung cell ka lang, in order for you to have a population, you have to divide yourself to create your own population. So mitosis is not only for growth and development of a single person, but also it's a way to increase of a single cell eukaryote to increase its population. So the end goal of mitosis, the very end goal of mitosis that I always want you guys to remember, is to create two daughter cells. The reason for cell division is to create two daughter cells with perfect full set of chromosomes, meaning the cell is diploid or diploid, or in other words, 2N. N talks about the chromosome. So in a single cell, there's 2N, one from mother and one from father. And if that cell has 2N, or two sets of chromosome from each of the parent, meaning they are diploid. So when a cell divides via mitosis, it creates two cells. And that two cells created, those two daughter cells, should still have 2N. They still should have two sets of chromosomes from parents and mother. Okay, I hope that's clear po. So cytokinesis is the division of cells cytoplasm. So mitosis is basically the division of cell inside. Cytokinesis is when you divide the cytoplasms to form two new cells. So again, it's just a continuation. Mitosis is you can also think of it as the division of the chromosomes and everything inside into two parts, one side and the other side. And then when you cut that one cell to make two cells, it's basically cytokinesis. Now let's talk about mitosis. So there's also four stages of mitosis: prophase, metaphase, anaphase, and telophase. Let's talk about prophase, the first stage of mitosis. In prophase, usually divided to divide pa rin sila into early prophase and prometaphase. So let's talk about early prophase. What is happening during early prophase, or basically the early parts of prophase? So what is happening is that it set the stage for division by breaking up and building structure. As you can see here, one thing na nangyayari is they starts to break up important, or it starts to break up unnecessary materials inside the cell, and it starts to build up structures that are very important for the division of cell. Moreover, chromosomes starts to condense. As you can see here, the chromosomes, at the beginning, they are very stringy. They're very like string structure. And these DNA starts to condense to create chromosomes. And these chromosomes, there's two pair of chromosomes in parang na nakadikit sila. And we call that sister chromatids. Moreover, mitotic spindles begin to form. Mitotic spindles begin to form from centrosomes. These are centrosomes. Mitotic spindles are microtubules na kumbaga para silang string na nade-develop from centrosomes to attach to the chromosomes or the sister chromatids. And that string na naka-attach dun sa centrosome, yun yung magpo-pull dun sa centrosomes para maghiwalay or dun sa chromosomes para maghiwalay sila. And the nucleolus also disappears. If you guys remember what nucleolus is, it's basically another central region na makikita nyo inside the nucleus. And kapag nawala yon, it's a good sign na nag-i-start na ng mitosis yung cell. So sa pro-metaphase part, chromosomes becomes even more condensed. So mas nagiging condensed sila, it creates a sister chromatid, and then magbe-break yung nuclear envelope. So yung membrane ng nucleus, magbe-break siya, it means marirelease yung mga chromosomes. And ang mangyayari after ma-release yung chromosomes, the mitotic spindles will start to grow even more and ika-capture nila yung mga chromosomes and organize them. So ano nangyayari? So these are the spindles. And these are attached dun sa tinatawag natin na centrosomes. Okay? So hindi lang nakakita dito yung centrosomes, but it's around here, and there's another centrosomes at the bottom. And these microtubules, this is spindle, are created from those centrosomes to attach to the chromosome. This is the chromosome, and these are two sister chromatids. And in the middle of those chromosomes, kung saan sila naka-attach, is merong kinetochore. Kinetochore is a protein on that centromere. So medyo maraming term, no? Centromere is basically the area that attaches these two chromosomes, sister chromatids, together. Yun yung centrosome. And in that centrosome, makikita mo yung kinetochore. Yung kinetochore is yung protein kung saan mag-a-attach yung mitotic spindle. I hope that's clear. So the centromeres are the region of DNA where the sister chromatids are attached, and it contains kinetochore wherein the microspindles will get attached. And then the microtubule spindles or the mitotic centrals of the microtubules or the mitotic spindles from centrosomes will arrange the chromosomes together, and then they will pull them apart. Okay, so that is what is happening in prophase. So let's move to metaphase. So in prometaphase, just a review, ang nangyayari or in prophase in general is that it breaks the nuclear membrane. It releases the condensed DNA, so meaning during prophase, the DNA are also condensed. And then the centrosomes starts to build up mitotic spindle, and then the mitotic spindles get attached to the sister chromosomes on the kinetochore. And the kinetochore is found in the centromere, and the centromere is the region of the two sister chromatids where they are tightly attached. I hope that's clear, and I hope it's not confusing at all. Okay, what is happening next in metaphase is that in metaphase, the spindle has now all captured the chromosomes. And ang ginagawa niya, they will arrange the chromosomes and align them in an area called metaphase. The metaphase is not an actual structure. It's just the central region wherein the chromosomes are aligned so parang ito. Nakita niyo sa moving pictures. This middle area is the metaphase plate, wherein all the chromosomes are aligned. And then, the two kinetochores of the microsomes should be attached na dun sa microtubules. So these are microtubules, these are centrosomes. Centrosome starts to create the microtubules, and then the microtubules attach to the kinetochore of the sister of the chromosomes. Before and then, after this, what happens? Before it starts to pull these chromosomes apart, because remember, nag-duplicate na yung cell, di ba? So there's twice the amount of chromosomes. So hindi lang ito 2n, this is already 4n. So they have to separate those to have 2N on one side and then 2N on the other side. And before it does that, again, it will have another checkpoint called the spindle checkpoint. This is spindle checkpoint. The cell will check if the kinetochores are correctly attached, or if the mitotic spindles are correctly attached on the kinetochore. If they are not, aayusin ng cell yan so that walang bad na mangyayari when they start pulling on the DNA. Okay, so that's just a metaphase. So the important thing to remember in metaphase is that the cells align at what we call the metaphase plate, and that the kinetochores attach the attachment of the microtubules to the kinetochore is double check. Okay, next, anaphase, PMAT. So that's how I remember these stages in mitosis. PMAT. Okay, so PMAT, PMAT. Next, anaphase. Anaphase is the protein acting as glue attaching the sister chromatids together is broken down, separating the two chromatids apart. So you can see here they're originally attached and the glue separating them at anaphase magbe-breakdown siya. If after it breakdown, magse-separate na yung two sister chromatids. So this set is 2N and this other set is 2N. And what happens is that the separated chromatids, which are now its own chromosomes, are pulled towards the opposite ends of the cell. So what happens is that the microtubules will start to pull on those DNA or those chromosomes towards one side and towards the other side. There's also some microtubules that are not attached to the chromosome. So hindi nyo lang makikita dito, but there are some microtubules that are not attached on the kinetochore. And they create a line. So if I'm gonna draw that, it should something like this. So from centrosomes, so merong mga microtubules that are not connected to the kinetochore. So what happens at this section sa mga to na hindi naka-attach dun sa kinetochore ng chromosome is they actually starts to elongate. So meaning they start to push this microtubules, they start to push the cells apart. So they start tawag dito, they start forming and getting longer and longer. And if you guys imagine, kapag meron kang dalawang stick, di ba? If there's two people pressing on a stick, they cannot go closer together. And then kapag start na humaba yung stick, mas nagiging apart yung dalawang tao. So that's how microtubules that are not attached on any of the chromosomes, ganun yung ginagawa nila. And then the ones driving the force of these process is called the motor proteins. So there are motor proteins that elongates these microtubules, and there are motor proteins that are pulling onto these chromosomes. Okay. And the last phase is the telophase. So the telophase, the cell is nearly done dividing. And then here it reestablishes the normal structures as cytokinesis takes place. Remember, cytokinesis is the division of the cytoplasm. So in telophase, it's almost done dividing, and then it starts to rearrange again. Lalagyan na ng nuclear envelope yung mga DNA, and then lahat na ng mga organelles mo nasa one side na, and then also on the other side. And then during telophase, you organize na lahat yan for cytokinesis to takes place, or for as cytokinesis takes place. And then also mitotic spindles are broken down. Two nuclei are formed. So those are the nucleus. The nuclear membrane are brought back. And then the chromosomes condense goes back to their stringy form. So hindi na lang sila condense na, they start to lose already. Nagiging string na ulit yung mga DNA. During either the anaphase or telophase, the cell also undergoes its second phase, part of M phase, the cytokinesis. So remember, this is the mitosis, tama? There's another part ng M phase which is called cytokinesis, which is the division of the cytosol. This cytokinesis starts either on Anaphase and Telophase. Okay, so cytokinesis happens either at the start of Anaphase or Telophase. Cytokinesis is the last stage of M phase where it divides the cell's cytoplasm, creating two new cells. So as you can see here, nahati na yung cytosol. So you have now two daughter cells. Moreover, it overlaps with the final stage of mitosis. And mitosis final stage is anaphase and telophase. Remember, PMAP, prophase, metaphase, anaphase, and telophase. And then ends shortly after telophase ends. In animal cell, a band of filament called actin contract, creating a cleavage furrow. So you can see here, there is a cleavage. So if we go back here, makikita niyo ba yon? Yung parang, I hope I can post this. I'm not sure if I can. Yeah, I can post it. But yeah, kung makita niyo, yun, parang may cleavage na yon. That cleavage is done by the actin. So these are filament called actin and they contract. And that contraction creates the cleavage furrow. And then the contraction keeps going and going. Kumbaga, if you guys have seen the video of a watermelon, they keep wrapping rubber bands in the middle until na mag-explode yung watermelon. So parang ganun yung ginagawa ng actin. Only hindi mag-explode yung cell, but it will divide the cell into two. So the actin will keep contracting until na mag-separate yung dalawang cell. In plant cells, though, hindi ganun yung nangyayari. Ang ginagawa ng plant cells, since stiff siya and meron siyang cell wall, it cannot divide by outside. It has to divide from inside to outside. So for cells, it kind of like divides from outside to inside, but for plants, it divides from inside to outside. And it starts by creating a cell plate in the middle. And then that cell plate keeps growing and growing until na mag-divide yung dalawang plant cells. And lastly, cell cycle and Geophase. So some cells divide slowly or not at all. So meaning after nila mag-divide once, tapos na sila. For those cells na nagdi-divide slowly, hindi lagi sila nagdi-divide, they enter a phase called G0 phase. In G0 phase, this is where the cell rests. We call it the resting state of cell cycle, or the cell cycle arrest state. So meaning the cell is preparing for cell division. But in G0 phase, what is happening is basically the cell is just doing its function. So it's functioning as it's supposed to. It's not duplicating its DNA, it's not duplicating organelles, it's not increasing its size. It's just there existing to do its job. At G0 phase, the cell is not actively preparing for division. Instead, they're just normally performing their day-to-day functions. Some cells have even permanent G0 phase and never divides again. So after they divide and reach maturity, they never divide again. Some of those examples may be nerve cells and cardiac cells. So nerve cells and the cardiac cells, so yung puso, after na matapos na yung maturity ng cardiac cells ninyo, yung heart cells ninyo, it will not continue to divide. That's why if your heart organ is damaged, it's actually quite difficult for it to restore itself. You usually have to have another heart transplant. And some cells naman, they just have to be reactivated to enter a G1 phase again and to divide by just proper signal. Okay? So that is our lesson for 5.1. So that is cells, cycle, and mitosis. If you guys have any questions, you guys can message me. If you guys don't have any questions, you guys have a good day, and thank you so much.